Next up, Georgia Tamaris's lab in DHVI has been hard at work doing developing novel assays to look at binding antibodies. And in Georgia's absence, Kelly Seaton has been designated to give us an overview. Kelly?
Good afternoon. I'm happy to represent Georgia's lab, the binding antibody analyses that we will be performing as part of the CFAR Immunology core. The focus of our laboratory is assessment of humoral response to vaccination or infection, either acute or chronic infection. And we do this through the use of highly customizable antigen-specific binding antibody assays in a Luminex platform, which I'll get to in a moment. We are also able to profile global changes in isotype or subclass responses to vaccination or infection. And the assays that we have currently available for HIV, TB, hepatitis C virus, and SIV, we're also open to other types of pathogens if anyone wants to collaborate on development. We've also demonstrated, particularly in the case of HIV and HCV, but also for TB, that these assays are very specific. So we could examine antibody responses in patients who are co-infected. The two main services that we offer are the binding antibody multiplex assay, referred to as the BAMA assay, and the peptide microarray. So the binding antibody multiplex assay is a multiplex capability, again, based on Luminex technology. We can assess over 100 antigens per sample, and typically we run panels of 10 to 30 antigens. We are also, as I mentioned, able to profile subclass responses, antigen-specific subclass responses. So IgG 1 through 4, dimeric IgA, secretory IgA, IgM, and IgA 1 and 2 as well. We have also recently developed an avidity index assay for the Luminex, which is a measure of the functional property of antibodies. Namely, we add a dissociation buffer to one well of the sample and compare that to binding in the absence of the dissociation buffer. And therefore, we can measure the relative avidity of antibodies against a particular antigen. It measures the maturation, the immune response in vaccination or infection. We also are able to perform total antibody analyses for mucosal samples. So often there is variability, particularly in cervical lavage or eluted samples from WEX in the amount of total antibody recovered. So we can perform total antibody analyses to examine antigen-specific activity based on recovery. We also offer, the second assay is a peptide microarray, which has cross-clade specificities, uses minimal specimen volume, and we're looking at HIV and SIV, GP120 and GP41 linear epitopes in a wide variety of samples. I'll go a little more into detail about the binding antibody multiplex assay, which was developed in our lab by Nicole Yates. This assay is a validated GCLP compliant binding antibody multiplex assay. We validated it in conjunction with the quality assurance unit here at Duke. We have, again, fluorescent Luminex beads, which we couple to the antigen of interest. We have both whole protein and peptides available to examine in vaccination or infection. These beads are then developed into a panel of antigens of interest, incubated with serum, plasma, or mucosal antibodies. We then have a detection antibody, and again, we have a wide variety of detection antibodies, various isoforms and isotypes. Every assay has a control, positive control, and often has a standard curve to quantify the concentration of antigen-specific antibodies in that sample. This is a very sensitive assay. We can reach nanogram per milliliter detection limits in our samples, and this is more sensitive than ELISA's. We found that in our lab. We can perform these assays on a multitude of sample types: mucosal specimens, saliva, semen, cervical secretions, rectal wax, vaginal swabs. We can do this from human or non-human primate samples. We have also looked at purified antibodies, monoclonal antibodies, and cell culture supernatants. So a very flexible assay for many different sample types that may be available. To highlight some work that has come out of this lab using this technology, Nicole Yates in our lab found that HIV-1 GP41 envelope IgA is frequently elicited after transmission. However, it has a short initial response half-life. So you can see in these subjects, we looked at both plasma and mucosal specimens, seminal plasma or cervical lavage from these two different patients, and you can see that the variability in the responses, or the difference in responses for IgG versus IgM or IgA. So again, we can look at global patterns of subclass or isotype-specific responses in infection or vaccination. Another study where we looked at HIV-specific IgG3 responses during acute HIV has implications for detection of incident HIV infection. So this table shows you that we looked at IgG3 responses to several different HIV antigens. And I know this is a busy table, but what I wanted to highlight is we were able to elucidate the effect of viral load only ART use or location, so clade A, clade B or C, and its impact on the antibody half-life in days. And we found some differences based on either ART use or not, or location, on the differences in antibody responses of the IgG3 subclass type. And we're following up on this work currently to work on development of an HIV incidence test in conjunction with the Gates Foundation. I'll now move into description of the peptide microarray, and this effort is led by Shauna Shen in our lab. And if you would like more information about this, Tong, who is an undergraduate in our lab, also has a poster here today. I believe it's poster number 25, if you're interested in taking a look at that. For the peptide microarray, we have an anti-envelope epitope mapping strategy, and this is for either full-length HIV GP160 or GP120 for 6 HIV strains, vaccine strains. We also have GP160 analysis available for 2 SIV strains. In this peptide microarray, we have 15 mer peptides that overlap by 12 mers, spanning the GP120 and 41 regions, printed onto a microarray slide, incubated with a complex protein sample, and then we come back with a labeled detection antibody, and we can then epitope map where these antibodies are binding on the envelope. This library has been designed by Betty Corbel at Los Alamos National Laboratories. And again, we have full-length consensus envelopes available for HIV, covering the major clades and CRFs. And we also have, more recently, 6 HIV vaccine strains, GP120s, that are in the, some of the more common vaccines being currently in trial. We also have available analysis of the GP160 for MAC239 and SME660 for HIV, and we have nearly 2,000 peptides available to epitope map these antibody responses. In the application of the microarray, we have both human and animal models, including rhesus macaque, guinea pigs, and rabbits. And again, just like the binding antibody multiplex assay, we have many different sample types available: serum or plasma, mucosal specimens, or purified antibodies. We can also look at IgG, and IgA is currently in development. The features of the peptide microarray: it's in place for human clinical studies, for non-human primate studies, and again, as I mentioned, small animal models such as rabbits and guinea pigs. We can identify immune correlates in vaccine trials in both humans and primates through this mapping. The peptide microarray can also map antibodies from, again, a variety of specimen types, so both plasma and serum, and also genital fluids. And it's a very flexible assay where we can add new peptides in our array library, adapt to other animal systems or sample types, and we're also beginning to look at segmenting our slides for decreased specimen volume and higher throughput. And some publications that have resulted from this technology, we were able to compare plasma and CVL IgG responses in a broad neutralizer from the Chavi cohort. And you can see here that you have differential specificity of antibodies found in the plasma versus the CVL. This was an interesting finding that was resulted from the microarray. We also did epitope mapping in the RV144 TIE trial, and identification of a novel epitope shown here the C1 IgA peptide led to the IgA envelope correlate of risk that was found in this TIE trial. In collaboration also with Sally Permar's lab, we isolated neutralizing mucosal monoclonal antibodies from human colostrum. And again, you can see by epitope mapping, most of the binding was seen in the C5 region for this neutralizing antibody. So we are happy to collaborate with anyone who has an interest in looking at antibody responses, either by binding antibody assay or by a peptide microarray. So for additional information for the binding antibody multiplex, you can contact Georgia or myself, and for the peptide microarray, you can contact Shauna. And in addition to CFAR, we have funding from several other sources, which I would like to acknowledge.
Very good. Thank you so much, Kelly.